So, you're a dental student, you just started university, very happy, and then suddenly this happens.
Coronavirus.
COVID case. COVID as a coronavirus.
COVID-19.
The second wave of coronavirus.
COVID-19.
The
COVID-19 infections.
Coronavirus.
Coronavirus.
Your lectures are cancelled, teaching is disturbed, and you're wondering, how am I going to pass these exams? What do? You watch my channel, that's what you do. Hi, I'm Nasser. I'm a practicing dentist from the UK, and on this channel I make content to make undergraduate life a lot easier. So, what we're going to start off with, I was thinking about a topic which I think a lot of undergraduate students might find a bit difficult or annoying or wonder why are we studying this. And there's no better topic to pick or start off with than biochemistry. It's one of them topics that nobody really likes studying, often overlooked, and you're often sitting there thinking, why am I studying this? And what is the problem with biochemistry? The problem with biochemistry is it's just so poorly taught. There's so many different textbooks, there's so many different lecture slides, and every professor seems to have their own way of teaching biochemistry. And it often ends up with a lot of students sitting there wondering, how am I going to study this, or how am I going to pass this subject? None of this is making sense. I mean, you open the textbook and suddenly you hit BAM with all these diagrams, all these pictures, and the explanations are either too vague or too deep, and nothing really seems to make sense. So often the advice dental students give each other is, all you think you have to do is just paraphrase learn it and hope for the best. Well, it's a subject I used to hate and I almost failed until I really looked into it a bit deeper. And I managed to really understand this subject and ended up getting 96% in my exam. And I've noticed biochemistry is not that hard if you just focus on a few important things. So what I plan to do is, I plan to go through a single pathway and give you a slightly different approach how to study biochemistry, which you can apply to all pathways. And hopefully you'll find it a lot more easier, you'll be happy studying the exam, and hopefully get a really high mark. So what I'm going to do is I'm going to switch over to my screen over here and show you guys how I plan to tackle this pathway. And the pathway I'm going to pick is obviously going to be glycolysis. Why glycolysis? It's normally the first pathway that every student's introduced to in biochemistry. It's probably a pathway you've definitely studied previously before university, and it's normally the one that links everything together. So what we're going to do is we're going to switch over to my screen and I'm going to try and make a small little presentation on notes which hopefully you can follow and copy along. I ideally wanted to get a textbook such as Lippincott Biochemistry and go through it, but I didn't get the copyright permission for that, so we're just going to freestyle this and see how it goes. So let's try and switch over to the screen. Hopefully you can see it, and let's get let's open a new presentation. I think that would be the best way to do it, and you guys can just follow along. Okay, let's have a look. Biochemistry. Okay. So what I'm going to do at the start is I'm just going to very briefly go through glycolysis. So I'm talking very briefly. I don't expect you to understand anything at this stage. Just so we become familiar with what we're going to be discussing. And then we're going to break it down for you. So let's have a look. Can we get Google images here? Yes, we can. Google images. And let's bring that across. Here we go. Hopefully you guys can see this now. And let's type in glycolysis. Search. Yes, here we go. Now, is your heart racing? Should be. Let's have a look. Let's find the simplest diagram we can get here. Is that simple? No, what use that? Let's have a look. Is that making more sense? That's it. Let's have a look at this. Very briefly, okay. Glucose goes to glucose 6-phosphate, goes to fructose 6-phosphate, to fructose 1,6-base phosphate or biphosphate, which splits into glyceride 3-phosphate, dihydroxytane phosphate, and it just goes all the way down to pyruvate. Okay, and what students normally do is write this out paro fashion, start from the top, right to the bottom, then learn the enzymes, then learn where the ATP is introduced or made, NADH, etc. And just keep repeating it until they go crazy. So no, let's not do that. It's a lot better if you try to understand this. Now you're thinking, oh, how am I going to understand this? Because you take loads of different approaches. So what is the problem here? The first thing we need to do is, and I'm talking about not just glycolysis, I'm talking about any pathway. The first thing we're going to do is, let's get that presentation back up. First slide, number 1. Okay, and I think there's around 8 points I want to make on this whole thing, but let's start with number 1. It's always ask the question, what is the purpose of this pathway? Very simply, and this should be in one sentence, one clear sentence, okay? Something that will give you an overall picture. Okay, this is why we're doing this. And it's very simply, we're gonna we're converting glucose into pyruvate, which either enters one of 2 different pathways, depending on if oxygen is present or is not present, also known as the aerobic or anaerobic pathway. Okay, so we're going to convert glucose, oh, look at my spelling, converting glucose into pyruvate, which then enters one of 2 different pathways. Look at my spelling. Depending on if there is oxygen present. Okay, but why? Right at the end, what we're trying to do here in glycolysis is obviously produce ATP, adenosine triphosphate. So let's put adenosine triphosphate, ATP. Right, and why we're doing that? Because ATP is the main energy function or source in the cell. Helps cell stay alive. It's a very broad term, but you know what I mean. It's used for enzymes, used from membrane receptors. Just to get everything good in the cell, you need ATP present. When there's no ATP, everything shuts down and you're pretty much doomed. The cell will go into apoptosis and die. Hashtag sad news. So let's have a look. So that's the pathway. Okay, so what is the problem with this? Is exactly what we did here. I typed in glycolysis, so all you saw was hundreds of different views of glycolysis. You got the simple view here, you know, just everything written out. You got something like this, all the chemical structures written out, along with the enzymes and everything going crazy with the arrows. You got, let's have a look at a different approach. Let's have a look at this here. Oh, let's have a look at this. Okay, I like this. I'll explain later why, but yeah, we're gonna like this diagram. I am a fan. Whoever made this, you are a legend. That's fantastic. Okay. So, what I'm trying to say here is, even though different views are confusing, one of the biggest tips I want to give you as point number 2 is you always look at different, let's put views of a pathway. Now the problem with buying a book is the author of that book or the team writing that book will always write the pathway which they think is the right way to do it. But there's always different ways of looking at this pathway, and sometimes looking at a different pathway at a different angle will make it a lot more easy to understand. This is why I don't think there's one good book for biochemistry. I think you need several different sources. So I'll explain to you why. So let's have a look. In the first diagram that we saw, there was nothing there. It's just a series of words. Nothing's really making sense. You can kind of guess what's going on. It doesn't even mention enzymes. Okay? Then you've got this diagram here, which is everywhere, everything is all written out. You've got the enzymes listed here. Okay? But nothing's really clear. Okay? And it doesn't motivate you, there's no explanation. I mean, here's a diagram. So, then the last one, the one that I like, where's it going again? There we go, this one. I think this is one of the best ones, okay? I like the way they split it up as well into two different phases, and that explains why. But let's go through this properly. Okay, so let's go through this and let's do a full explanation. So what is a biochemical pathway? I mean, I'm just gonna digress here slightly. Okay, it's a series of reactions where the product of one enzyme or one reaction is a substrate of the following reaction. So it's like a chain reaction that happens, and the best way to think of it is a bit like, I'm not sure if every guy's ever seen a factory or assembly line. So let's put into a factory assembly line. Let's see if we get a nice little picture video here somewhere. Assembly line, no. It'd be better if there's a food assembly line. Can we get an assembly line here? There we go, almost. Yeah, I like this. There's a possible assembly up there for that. Let's have a look. Let's forward this. Don't waste no time. Here we go. That's more like it. Something like this. So these guys are packing boxes, right? And the line's moving across it. So, and everything has to stay in order. So the boxes that they're packing, imagine it's glucose. Okay, it's going to the first enzyme. He's modifying it. It goes along the line. It goes to the second enzyme. He's further modifying on the line, third one. So you just like that. Oh, let's turn that volume off. There we go. Does that make sense? So that is basically a biochemical pathway. Okay, so it's a series of reactions that happen where the product of one reaction is a substrate of the following reaction, and everything has to happen in order. Now let's have a look at what happens in glycolysis. Okay, and let's break it down, and you should always, right, so what I'm trying to say, let's put that down. Let's get some complete notes for you guys, just so everything's clear. I don't want to just ramble on here. Think production line. I think you guys know what I mean now. Let's put assembly line in a factory. Product of one reaction is the substrate of the following. Okay, perfect. Next point we're going to make, and we're really going to start doing this in a bit of detail now, as in, like I said earlier, nothing is random. Each enzyme has a purpose. Okay, so let's break down glycolysis now. Okay, let's have a look, because what we need to do is, let's go through it truly now. Which one shall we pick to use here? Should we pick this? Yeah, let's pick this. I like this. So glucose here, 6 carbon molecule, right? What's happening to it? Basically, glucose enters the cell. We'll go into detail in a different video of how it enters the cell with special transporters. So glucose enters the cell and it meets its first enzyme, heterokinase. Okay, heterokinase uses ATP and turns that into ADP, and it turns the molecule into glucose 6-phosphate. So very simply, let's break it down. Okay, glucose enters, a phosphate group from the ATP is being transferred over to glucose, and it's called glucose 6-phosphate. Always remember, in these pathways, everything has to be logical. Nothing's named randomly. So a lot of the time, in the title itself, it gives you clues what's happened. So very simply, look very carefully, glucose 6-phosphate, basically the phosphate group has been added on the 6th carbon molecule, 6th carbon on that chain of that glucose, so it's called glucose 6-phosphate. There's a phosphate there now, where it's come from ATP, right? So glucose 6-phosphate turns into fructose 6-phosphate by the enzyme isomerase or phosphoglucose isomerase. Now, which then moves forward and it goes to fructose 1,6-biphosphate. So what's happening here is, very simply, glucose is getting a phosphate group added onto it on the 6th position, 6th carbon, which then is using an enzyme called isomerase. You know, remember isomer from organic chemistry, like a mirror image. It's changing the shape. It's changing the shape of the molecule. So it goes to glucose 6-phosphate to fructose 6-phosphate. So the phosphate group is still there. It just changes the chemical structure, arrangement of this molecule. And why is it doing that? Well, what we need space for the next phosphate group to be added on. Now, a little side note, why are we adding phosphate to glucose? Just a random question. Why do we add phosphate to anything in a cell? Now, that brings us to the question of ATP. ATP is the energy source, right? Phosphate, the phosphate group on the, let's open a new window here, let's have a look. No, very good, let me just right here. Let's have a look. ATP, here we go. Images. Right, here we go. That's what I wanted to see. The adenosine triphosphate, right? It means there's 3 phosphate groups on the molecule. The last phosphate on the ATP, the energy bond there is very high. What does that mean? That means it's very easy for ATP to pass that phosphate group to another molecule. That bond is very easily broken. When the phosphate group is added to another molecule, it gives it energy. So the whole idea of glucose turning to pyruvate, what we're actually doing is, or what the cell's trying to do is trying to give it energy. Why? It's trying to break it apart by adding energy into it, as in making it big, very kinetic, so it splits into 2. So it's added this first phosphate group on the 6th molecule here, 6th carbon here on that glucose. Then it's changed its shape and it's made space for another phosphate group to be added there, which it does. So fructose 6-phosphate goes to fructose 1,6-biphosphate by another enzyme called phosphofructokinase, which adds it to the first position, the first carbon in that chain now. So therefore, you can understand why it's called fructose 1,6-biphosphate. The 1,6 is mentioning exactly where the position of the phosphate groups are on that molecule. Now, you've got 2 phosphate groups added now to 1 molecule. It's going to make it very, very unstable now. It's giving that molecule a lot of energy. Now, what's going to happen now is another enzyme comes, walking past, aldolase, and it's just going to split that molecule into 2, which are almost identical. Now, where's the other diagram gone again, which we were looking at before? But that showed it really nicely. There we go. That is fructose 1,6-biphosphate, literally split through the middle-ish, and you've got the 2 products of that glyceride 3-phosphate and dihydroxyacetone phosphate, right? And this is where you have to pay really close attention. Both of these cannot move forward in the pathway to become pyruvate. Only one of these can. And where's that molecule done before? Only the glyceride 3-phosphate can. So now you're splitting the molecule into 2. You basically, it's very, very unresourceful to only use half of that. So the cell does need a way of turning the product that can't continue the pathway into a product or glyceride 3-phosphate which can. Like I said before, the enzymes are very specific. They only going to attach to a substrate which it recognizes, and the next enzyme in that whole production line is the glyceride 3-phosphate dehydrogenase. It will only recognize glyceride 3-phosphate. So the cell has another enzyme called triisophosphate isomerase, another one, isomerase, as in a mirror image, one that changes the structure slightly, or arrangement, so it turns back into glyceride 3-phosphate. Now, what do we need to add on to our presentation here? Every enzyme has a purpose. Yeah, which, okay, let's go to number 5. Is it 4? Yeah, number 5. Now we need to do something else. We need to pay close attention to everything. And what do I mean by that? I mean, right now we're focusing on what's happening to glucose, right? And we're focusing on what enzymes are involved. One thing we're not focusing on, which we should be, because it's a very important concept in biochemical pathways, and often things that pop up in the exam where people overlook and then they get caught out, is, let's have a diagram that shows it properly. Here we go. I like this one here. You've got these arrows here which are basically saying is a reversible reaction or not. That step, glucose to glucose 6-phosphate, can't be reversed. But this one can. Can't be reversed. This one can. 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 This one can't. Now, it's very, very important to pay attention to these, because it's a very easy exam question asking, is so and so step, is it a reversible reaction or is it an irreversible reaction? And you always need to wonder, why are certain steps reversible and why are certain steps not reversible? So my point over here I'm trying to make is, always pay attention to if a step is reversible or not. It's an easy exam question, and will very likely be asked in every exam. Yeah, okay, let's move on to the next slide. And this is a very important point I'm going to make is, you know the pathway, you know what's happening, you know the purpose of the pathway. Now, there's something which will always be asked in the exam, but it's something for some reason that's not really emphasized enough, I find anyway, in books or in lectures, is, have you ever seen that cartoon, Where's Wally? Where's Wally? Or Waldo, if you're in America. Where's Wally? You find this character, this strange looking character. You get a diagram, you need to find them somewhere, right? And your whole focus, you pay your whole focus to this until you find you get really happy. I want you to be in that mind frame when you find or ask this question. Find the regulatory enzyme, or even the enzymes. What are the regulatory enzymes? Now, if you think of this as a production line, right? There are certain enzymes, or people, if you use that analogy of assembly line, which control this pathway, which let him move forward. Now, you can probably already guess, is it the reversible ones or the irreversible ones? It is the irreversible steps. The enzymes there, heterokinesis, where we go, phosphofructokinase 1, pyruvate kinase, right? They are irreversible steps. These control if the pathway moves forward or not. Now, if these decide, or if these enzymes decide the pathway doesn't move ahead, the whole thing stops. Glucose stops being produced into pyruvate. So, what are the enzymes? They control the pathway. Now, the question to ask is, what regulates, before then, which enzymes are they? All of it list these, right? What enzymes are they, and what controls these regulatory enzymes? Because obviously they're not just sitting there making their own random decisions, and they all need to be in sync. For example, one can't decide, yeah, we'll go ahead with this pathway. Other one says, no mate, you stay there. Other one says, yeah, I'm confused, please tell me what to do. We let your third friend, I'm sorry with you, doesn't know what's going on all the time. It can't be that everyone has to be in sync. Now, that means they must get their orders like a general from somebody higher above. So there's things that tell these regulatory enzymes, yeah, it's a go ahead, or no, let's stop. So either these are local factors or they're systemic factors. Now I'll give you an easy example. Let's have a look. Yeah, for example here. Okay, let's pick the first one, heterokinase. Glucose to glucose 6-phosphate. Heterokinase uses the ATP and gives it to phosphate on the 6th position. Now, glucose 6-phosphate actually inhibits heterokinase. Why? If you think about it, if glucose has entered the cell, it will only let it move forward once that glucose is uptaken by the next one. So, as soon as glucose moves in, it stops after it's produced. As soon as it goes into fructose 6-phosphate, it is no longer inhibiting heterokinase, the glucose moves in again. Now, if there's a high production or high concentration of fructose 6-phosphate already there, it won't let that glucose 6-phosphate move forward. So therefore, that stops. I'm hoping I'm making sense here. Right? So, local factors control the regulatory enzymes, but more importantly is the systemic ones. Now, let's see if we can get a diagram. Okay. Let's drag that across here. Let's open that up. Here we go. Okay, so never mind glucokinase here. It's the same function as heterokinase. Glucokinase is in a different set of cells. We'll do a different video about that. Don't get confused about that. Just think of it as heterokinase. Okay, insulin turns on the pathway or tells the regulatory enzyme to continue. Again, insulin tells it to continue. Insulin says continue, and is evil twin glucagon says stop, stop, stop. Right. So everything, one order here is the same as the rest of them. They all get the same instruction. Now, if you think about it, why would insulin tell the heterokinase or glucokinase, yeah, continue this pathway? Because obviously, what happens when insulin is released? It means the body has had an intake of carbohydrates, had an intake of sugar. So what's happened here is glucose is going around the body, it's entering the cell, and it's telling the heterokinase. And how it's telling the heterokinase is another pathway, which we can go through in a different video. But the end of that pathway, it gives a signal to the heterokinase to continue. So this is a very easy exam question where it says, which of the regulatory enzymes of glycolysis is definitely being the exam. Regulatory enzymes for every pathway. If there's a pathway mentioned, they will ask about the regulatory enzymes, and then they will ask what is influencing these enzymes. It's a very, very important point. Every pathway you study, look for the regulatory enzymes straight away. I mean, not straight away, but as part of your studies, always ask what is the regulatory enzyme or enzymes, and what is influencing them, therefore what is influencing the pathway that I'm studying. So let's move on apart from that. Yeah, so back here again, let's have a look. So hopefully that makes sense. So these are the points. We're find the regulatory enzyme. And the final piece is number 7. Put it all together. Once you get all the information, either go through the whole pathway with a diagram. Well, find the diagram that, in my opinion, is something like this. It's a really nice diagram, this, because it explains everything clearly. First, it writes everything out for you. It shows you if the reaction is reversible or irreversible. It tells you which enzymes are involved clearly on the side. It shows you the chemical structure and how it's changing step by step. And if there's an essential step where the molecule is breaking, it's showing you how it's broken. And it just breaks everything down quite nice and neat, and shows you where the ATP is used or where it's produced, or the NADH. That is a very nice diagram. Yeah, so put it all together. So gather all the different views of a pathway. Ideally find the one that I just showed you with everything on. Okay. Find the regulatory enzyme. Always ask why, as in, why is insulin affecting this pathway? Why is glucagon stopping this pathway? Why is phosphate? Why is the glucose being rearranged to make space for the other phosphate? And that's it. Now the final thing I'm going to leave on is, which question that people always ask is, should I buy the book? Buy a book or books for biochemistry? There's no right or wrong answer for this. It's basically, it depends. It depends. I think a lot of the structure, I think you do need one good textbook, just so you can read through it and study and give you the basic foundations of a pathway or pathways, or the whole subject. And the book I normally love, or have been loving, is the Lippincott illustrated book. I'll put a link somewhere so you guys can find it easily. But the rest of it, I think, just like I did right now, it's all on Google Images, guys. And so many different people put pathways on different ways. There's loads of videos on YouTube. Well, like now, there will be another one. Hopefully you're successful. Any questions, please leave it in the comments. I'm very happy to answer questions. And I wish you success for your exam. Now, that's about it. Good luck, guys. If there's anything I can help you with, give me a shout. And that was it. If you like this video, why don't you give it a thumbs up and subscribe, and watch my other videos and see what you think. And for now, thank you.